Our next speaker really needs no introduction. We all know who she is. We love her immensely, and we love the speaker after Sandy. So, Sandy, come on, Dr. Sandy Buckmaster.
Okay, come on, pick a side. It's like that when I drive too. I'm from New York, so everybody's always in my way, no matter where they are. Has this been an amazing NAIA conference? It's just incredible, and the speakers have been just amazing. They've been entertaining, they've been informative, and it's really been special. Just to point out, I belong to several research advocacy organizations, Americans for Medical Progress, of which I'm chair, Texas Society for Environmental Research, I'm president, our executive director, Lorraine Huff, was married. Lorraine Hayden is a huge fan of NAIA, and is how I learned about NAIA, actually, because she came and spoke to Patty, and then became a member of the NAIA board. And this, to me, has been incredibly special because of all of these organizations of which I am a part. This is, in my opinion, the only real, true umbrella organization out there that has the capacity and the will and the experience and the credentials to bring us all together, join us in our independent platforms, and pull that together in a form of synergy that really will have an opportunity and an impact on worldviews. So to me, NAIA is the be-all, end-all. We've heard about all these different topics we've covered. Right? We covered a whole bunch of things yesterday. We covered many more today. We're going to talk about some others, in fact. And I know Patty had mentioned the various platforms. Just to give you an idea, within NAIA, we have the Homes for Animal Heroes platform, which is my favorite one. But we have the Shelter Project, and there are numerous others in the works, and Patty's going to share that with you later today. And what I want you to be thinking is, and Michelle just, Farm Babe, sorry, just told us, hey, get out there, you need a platform, right? We need platforms, and we need to get on social media. And that's great, we should all start talking, but we need a place to talk from, a place that people know about, that has a following, that has a huge following, and that's NAIA, and we're about to create platforms for you. So, there you go. That's my NAIA plug, because I love it. And now I have a question for you. Medical breakthroughs. Is research with animals still necessary now? Yeah. I'm going to ask you a question, and I want you to be very honest. Have you asked yourself this question because you're really wondering if it's the case? Raise your hand if that's so. Sure, that's right. I mean, I have too. Yeah. And I mean, look, I'm not going to be upset by this. That's right. Have you asked yourself this question? I know you have, and you're just being shy because you think I won't like it if you say. But I will. I want to know. And it's actually very, very relevant because just as the Farm Babe pointed out, there's a lot of misinformation out there and a lot of information that the non-experts are sharing with the public who know very little about science. And I suspect that many of you will start to understand what's really happening in biomedical research and how it's structured in a way that you hadn't prior to this talk, which is why I put it together, okay? So let's start looking at it, right? I mean, to begin with then, we really sort of need to break down how to turn this on. We really need to break down what do we mean when we say the word research. People throw this word research around all over the place, and you may remember that yesterday I asked Diane Brown, Dr. Diane Brown, to separate basic from applied, and I think that was probably in and of itself a little confusing for folks because you don't really know what that means. People throw the word research around, and it means a couple of different things depending on what it is you're looking at and what your goal is. But that's never clarified for the public, and so images and goals get conflated, and sometimes we're not always talking about the same thing. So let's look at how research sort of works in general. Biomedical research is actually a continuum. It's not this thing with an official start and end. It's not like we're going to do research on that thing, and then when that thing is here, the research is done. This is just not how it works. It's a continuum. And it's a continuum that is comprised of two major parts, and I'm trying to simplify this, right? Something we call basic research, and you can see how gigantic that is. on purpose, and it has an arrow because it's ongoing. And then something called applied research. So what does that mean? Basic research is all about learning and understanding complex living systems. And this is complicated, so we have to learn about them and understand them on many levels, on gross levels, on microscopic levels, on molecular levels, and they're very, very complex, right? We may have to, it depends on the question you're asking, and there are many, many questions, right? But there could be millions of different questions you want to ask about all of these various things, depending on what part of health you're studying. And there could be millions of people working on answering some things within those levels. And then there could be cell cultures involved in that study, and there could be computer modeling involved in that study. But at the end of the day, the only way to understand complex living organisms is to study complex living organisms. And here's an example. Back in the day, and actually, I'm really glad that most of you in this audience will know what I'm talking about, because usually when I say back in the day, like I just gave this talk to a high school class, they had no freaking idea what day I was talking about. I couldn't imagine anything like what I was saying, because it was so, like, what are you talking about? But, okay, so like what day? So back in the day, if you were driving a car and it started making a little knocking noise or something, right? You went to your friend or your father or a mechanic, and what they did, they opened the hood, boom, and looked in there, and they looked around, they touched a few things, and I know exactly what it is. And then they go in and they make the correction, and that was your repair. Let's think about what was really happening there. These people knew exactly how to recognize what was wrong, because they knew exactly how that engine looked when it was functioning properly. They knew all the parts, they knew how they communicated with each other, how they interconnected, what did what. They understood the health of this engine. And by understanding the health of this engine and how it all works when it is functioning properly, they're then able to recognize, well, what's off? What went wrong? Right? And then once they figured that out, they could create a solution to repair that issue and bring that engine back to health. So think about basic research sort of like that. In order for anybody to recognize a disorder or what the cause of a disorder, they have to first understand on all of these very complicated levels everything there is to know about how that system functions when it's healthy, just like an engine. Once you know that, you can identify, well, there's something wrong there, and we heard some of that yesterday when we listened to talks about genetics from Dr. Brian, right, and a couple of other folks. Oh, there's something wrong there, and then eventually maybe we can sort out how to fix that, right, and then create health again. So that's what basic research is about. It's really about understanding living systems. And what we've heard countless times already in this conference is we have barely scratched the surface when it comes to understanding how even small parts of complex living systems actually function on all of these various levels, right? And so people will say, for example, and I like this when they say this, you know, you don't have to do this research with animals, these complex living systems, because you can program a computer to do that. And how many of you have heard this? They say it all the time. But remember, how much do we actually know about these very complicated living systems? Very, very little. It's extraordinary to me when I think about how little we know and how far we've come, right? We just heard about how we can't make enough food because people are living so long. I suppose that's our fault. But it's extraordinary, really, right? So what I like to say to these folks when they say the computer thing is, well, let me just think about this now. What you're saying is that you would like me to program a computer to behave in every way and on every level like a complex living system. And I don't really know hardly anything about complex living systems. So what you're suggesting then is that I program a computer to behave like something I don't fully understand in order to understand more about what I don't understand. This sounds like a great idea. Let's do that. And this is sort of the crux of basic research. Now, I would say the animal activist folks, what were they calling? Our special friends. Yes, our special friends. I hope I pushed the right thing here. Let's see. I did not. Let's see if this is it. No, there appears to be none of those things. I got it. And there it is. Okay. So our special friends, they like to refer to this sitting with healthy organisms and then maybe diseased organisms, or trying to emulate or replicate disease. They like to call that idle tinkering. And that's what they tell the public basic research is. These are people that are just curious about stuff, they're getting a lot of money. There's no point to this, it's idle tinkering because it's not going to become anything, right? And the truth of this, if you look here, is that basic research, this trying to understand these complex living systems on all these various levels, started in the 4th century BC. With the Greeks, naturally. And they had a lot of time on their hands, the Greeks. They was doing all kinds of stuff then, right? But you'll see that I've drawn it out to what looks like an infinity here, because I can't imagine a day when we'll really understand all there is to know about complex living systems. I mean, it's just, we're a part of this large fabric, right? We might be one little thread, and that thread's trying to understand something it's just a part of, a tiny part of, right? But yet we continue on. And it's because of that, then, that eventually there's enough clues and I'm talking about all over the world, right? There are millions of researchers doing basic research, trying to understand all these things we talk about, all over the world. And they're working with millions of animals, complex living systems, and they vary depending on what kind of system they're looking at and what their actual goal is. And they're all writing papers. And all this information is getting dumped into places where people that do science can find them, right? And there's just this gigantic pot of information, and it accumulates and grows and grows and grows and grows. And then at some point, somebody takes all that information and they see that there's enough clues in there for them to maybe come up with a way to fashion something that might repair that broken engine, right? And that's when we get into this next part of research, which is applied, right here. So now we're talking about, drug development, vaccines, new surgical techniques, new diagnostics like MRIs, PET scans, all these things are a result of basic research, these things we take for granted, right? So that's the applied part. And when you have thought about research in the past, be honest, was it not that part that you were thinking about? Raise your hand if that was the part that you were thinking about, the part that ended up in some kind of treatment for you, of course. And this is what the public thinks about. And in part, they only think about this part because they were told that this part is a waste of time, it's idle tinkering, and has absolutely nothing to do with what happens over here. But everything that happens here is fed entirely by decades and decades and decades of information fed by this gigantic, ridiculously huge arrow. I couldn't make it big enough. This never happens without this. That's a fact. Now, the applied part, now we'll talk about where all this is happening. This is where let's take drug development, so we can stick with one example. This is where people will take all those clues that they've gathered from all the basic research that's been done for decades, and somebody, let's say a pharmaceutical company, whose job it is to evaluate these things, will say, hey, you know what? There's a lot of good information here. I think we've got enough that we might be able to create a drug to model whatever's missing in the system, right? And we just plug that baby in there, right? We'll make it very simple. Now that work will take about 10 to 15 plus years, depending on the drug and depending on the nature of the study and depending on how many people actually have the disease, right? If only a few people have the disease, it's going to take longer to flesh through. And we'll talk a little bit about that now, right? So let's look at the applied part, the drug development part in our example. So you got this substance finally, right? You made this little molecule, you decided, I think this baby's gonna work, right? Well, now what we've got to do is we've got to test it to see if it works. And that brings up the other term that is often used incorrectly. This is basic research, and this is what people commonly call testing. Now what we need to do is test this molecule we've created based on all this information we've been collecting for decades, and see if it's actually going to do the thing that we hope it would do, like fix the engine and make it run again, right? Now there's two major phases to that, stages I should say. There's the preclinical testing, and I have that here in purple, and this involves animals. So, to recap, in order to understand living systems at all, we needed what? Animals. And people, depending on how we're asking these questions, right? But we need complex living systems in order to learn the things we don't know, right? And then when we get to testing, we need animals in this sort of preclinical phase. So what's happening there? Well, the first thing that people need to know, and by the way, there's a particular group of people known as the FDA that say it's the law for you to do this. This is not optional, so people need to know that too. The law requires that before we test a little wonder drug in human beings, that we find out a few things about it in animals first, right? So they want to know a couple things. First, is it safe? You know, is it toxic to the animals? You know, are the side effects, is it destroying various tissues in the animals and organs, and is it just not going to work, right? We don't know, right? And they want to know that too. They also want to know if it's efficacious. And don't confuse this with effectiveness, and this confused me for a long time. What this really means is they want to make sure that if you, for example, are created a drug to treat some liver disease, that you actually find some action in the liver. Right? So otherwise, then those drugs get thrown out and they go back to square one. Okay, so that's the animal part. The animal part's pretty short relative to what happens next. So that's it. It's really just testing. Does it go where it needs to go? Yes. It seems safe, and I will tell you that there have been less than a handful of instances where this sort of screening for safety before putting these molecules into humans has not worked. It's almost always effective in this regard. Just a couple, right? If the thalidomide was one, there was one, I can't remember, just a few years ago that a couple of children died in clinical trials and they had to pull it off. But for the most part, this preclinical phase with animals works really well at screening compounds to ensure that these little wonder drugs that we think we are so smart that we created will actually be okay for people to take. All right, so then we get on to the longest part of the applied research, of the testing. And that's the clinical trials. Now, the clinical trials all happen in human beings. The animal part is tiny. The bulk of that work now is in human beings. So it happens in 3 phases. I'm not going to go into all of the details, right? But it's 3 phases of clinical trials with different populations of people. And like I said, this can take up to 10 or 15 or more years. But now we're looking at safety, efficacy, same things we looked at before, dose levels. But then we also want to know, if we follow people out, you know, after, let's say, several years, depending on the disease, you know, what's their whole constellation of symptoms look like? How do they feel? And you can't ask the animals these questions, right? You can test blood to see what enzymes change and all of this, but in order to really know if the drug's effective, we've got to draw that out over some time and do sort of a long-term study. Sometimes it'll be short. There are a lot of different reasons, and actually, Dr. Mack is going to share with you sort of an exception to this rule. They do exist from time to time. So that's basic research, and then testing. We agree that animals are necessary here. Yes. Why? Because we don't know what we're talking about yet. We have to learn a lot of stuff. Right? Good. We agree that they're necessary here. Why?
Testing to see if it works.
Testing to see if it works. Why else? It's the law.
Oh, my law, yeah.
FDA said you had to do it. It's the law. Okay, now where does this stuff happen? You look at these 2 different kind of branches of research. Where does it really all happen? Basic research, for the most part, is funded by the government. Just to give you a kind of a little hint. To produce one drug, now I'm talking about, like, let's say over here, it could cost like $1 billion for one drug, maybe more, right? So imagine then how many billions and billions of dollars it takes to fund people in the United States who are looking at all of these other things, many more billions, right? So the government funds most of the basic research that's happening. You get some help, like from organizations like Canine Health Foundation for various specific things, and maybe some other disease organizations, right? But for the most part, the government is funding this. It goes on for decades and decades. Tell me how many pharmaceutical companies have the money to pay all of these people to learn about things for decades before they finally create a drug. They do not. There's no such thing, right? It's kind of impossible when you think about it that way. So you'll have this work happening at government agencies, like at the NIH or the USDA or CDC, for example. Mostly you'll find it in academic institutions that are all over this country. And they're getting funding, mainly from government agencies, to do this really critical work about understanding the complexity of living systems at all of these various levels. If you can't fix an engine, you don't understand, right? It's as simple as that. Where is the other, the testing end happening? We're talking about pharmaceutical organizations in this case, the early stages of drug development and testing, something called contract research organizations. And this is where we're doing a lot of this preclinical and then clinical trial stuff too. Hospitals, clinics. So to give you a sense then of how research really looks and where it's happening, and what the various focuses are, I created this slide, and it took me like an hour to create this one slide. So I was trying to figure out, how do I explain all this in one slide? Are there any questions about this? Because it's sort of important that you understand. Raise your hand if you've just learned. Yes, a question.
How much research is done on, say, rats versus...
Good question. How many animals in research, what's the breakdown of animals in research? It is over 97% of the animals in research are rodents, primarily mice. That means that 3% is everything else, and less than 1% are dogs and monkeys. So a very, very, very tiny fraction. In the VA, a couple years ago, there was all this discussion about dogs and cardiovascular research, and the number of dogs that were part of the VA system during that time were less than 0.05%. Less than it might be 0.5%. I think it was 0.05%, but anyway, tiny bit. Okay, so that's a good question, thank you. I was going to ask you something else and I've forgotten what it was. So,
Shame on you.
Shame on me. Well, I'm getting older. And my brain is full of such great information, I'm getting a little confused. All right, so this is how it's sort of all put together. I know what I was going to ask you. Now, how many of you, raise your hand, have a better understanding of how it all sort of works and fits together? Great, and why animals are so important still and critical. Exactly, all right. So here's what happens, right? Decades and decades, maybe another decade or two, and then this is what happens. Here's a great example of what happens. And that's, oops, hang on. Oh my God, okay. Jeez. All right. At least we get to hear that catchy tune again. How many have heard of immunotherapy to fight cancer before? Raise your hand. Oh, you're such smarty pants. My God, I guess because you read the news. Because these two gentlemen won the Nobel Prize for this technology this year, right? So, in brief, T cells, which are part of your immune system, go after invaders. Cancer's an invader, goes after it, tries to kill it. Cancer's really clever, and it stops it in its tracks, so it can't completely kill it because of these sort of checkpoints that get manipulated. And then these clever guys figured out how to disarm that checkpoint and allow the T cells to continue killing the cancers. And the beauty is, because it's a part of your immune system, if the same cancer comes back, you're just like a booster, right? Just like a vaccination you get. It'll go after the cancer again. It's pretty extraordinary. This is an amazing technology, and I'm not exactly sure which particular cancer they were targeting initially, but you saw at the end that now they were going to be able to target many, because once you develop a technology, which came from years of basic research and understanding, right? And then some of that applied research, once you have a technology, you have it. And now you can think of all kinds of clever ways to use it, right? This is how research works. And this is why animals are still so critical. Now these two gentlemen, just to make my point, won the Nobel Prize, and this is James P. Allison, and he is at MD Anderson, which is where I took that video from. And then Tetsuko Hanjo in Japan. I have a quote here from James Allison, mainly because I would not be able to understand the quote from Tetsuko in Japan. But I want to read it to you. It's actually very important. It says, "I never dreamed my research would take the direction it has. It's a great emotional privilege to meet cancer patients who've been successfully treated with immune checkpoint blockade. They are living proof of the power of basic science, of following our urge to learn and to understand how things work." Does that sound, and does that thing I just showed you look like idle tinkering? No. That's exactly right. Now I want to tell you something else about James Allison. It was never his intent to study cancer. It wasn't even, he never even thought about studying or treating or curing cancer. This was a man who was purely interested in learning about T-cell biology. It was all basic research. And while studying everything there was to know about T-cells and how they worked, he stumbled upon something that he then realized, because of the basic research other people had done, would have some utility toward cancer. That's how it works, right? So when they say, oh, this basic research doesn't lead to anything, they mean it doesn't lead to anything tomorrow. We don't know where basic research is going to take us. The more we learn, the more clues we get, the more we start figuring out how to connect those dots, the more likely it is that miracles will happen. So, you'd think, with something as powerful as this, that people would support this work more than they do. Now this is one of the polls, one of the recent polls. I think it might be Pew, it's a Gallup poll, okay? And I don't know if you can, I think you can see what's on here. I think it's real funny. And so the question is, you know, is this acceptable to you, to the public? Is this acceptable to you, right? So people are pretty okay with birth control and divorce, and it goes on down the line, right? Extramarital affairs, that's the least acceptable thing. That's pretty funny given how the world actually works. But the thing I find most striking here is, I like this, here, the death penalty. It's actually more acceptable to kill people than to save them. Right? Fascinating. More acceptable to kill people than to save them. This is sort of what's happening in our world. And why is that? Of course, everybody knows why. You can tell me, why is that? They're getting this information from the special people, right? We've heard countless examples of that against ag, against greenhouse emissions. Well, I guess that was ag, right? So I'm just going to give you this quick example because Patty asked me to. It's happening right now. So in research, and I've heard now many examples of why dogs are very powerful models of study. Primarily that's because we get many of the same diseases. We figured out ways to help each other in this regard, right? But I will let you know that there are only 3 major Class A producers of dogs for us. These are groups who breed these animals expressly for the purpose for us. They're not pets. This is important because we have pedigrees. We know a lot about these animals. We know all about their health. There's very little variability that we don't know about. That would not be true if we used people, by the way, who have all kinds of variabilities and have had all kinds of illnesses and diseases and then lie to us about what it is they have, right? So, and depending on what you're looking at, it would not work with dogs from shelters or pounds, right? So these are animals that we have a lot of information about, and that means that when we ask our questions, we're pretty certain about the slate we started with, right? We won't get confused then about what it is we may find and whether or not it's real or something related to the variation in these animals. Which is not to say, of course, that animals aren't varied, because they are. And they're also not a perfect model for the study of human disease, but neither are people for the reason I just mentioned, right? So there's only 3. And there's some animal rights folks out in Wisconsin right now, one of them's in Wisconsin, who infiltrated this facility, took some videos of dogs living, healthy dogs, and then intermixed it with some other videos that had nothing to do with this facility, and then created a story that really, much of which has nothing to do with this facility, and put it out there and got the public really wound up. And what they're doing is they're calling this a puppy mill for research dogs. So they're getting a double whammy. They get all the baggage from the puppy mill movement, and now they have all the baggage from the research movement. So they're going to get handily crushed if we don't do something about this, because the community is in uproar. How much of an uproar? Here's the really amazing part. On November 6th, they are proposing a change to the municipal code in this area to ban the practice of breeding research dogs, to ban the practice of doing research with dogs and cats, and they're putting it under their public nuisance code. The modification would be to their public nuisance code. This is what's amazing to me, right? So I'm going to read this to you, right? So they list there's 4 already, and then they're going to add the 5th. The following acts, omissions, places, conditions, and things are specifically declared to be public nuisances, offending public morals and decency. And this is what's on there so far. Gambling devices, the unlicensed sale of liquor, continuous violations of village ordinances. I mean, like, I don't know, running around the park naked sometimes or something like this. Illegal drinking, running around naked and drinking in the park, because you're supposed to not be drinking out of. Right, so those 4 things: gambling, illegal sale of liquor, doing something you shouldn't do in public, drinking in public, and saving lives. Yeah. And you're laughing because it's so ridiculous, but it's not so funny because these people think that this classification is completely appropriate because they have been so misinformed by the animal rights movement about the truth that they really view it this way. Offends morals and public decency. You think those people that are going to benefit from that therapy that the Nobel Prize winners developed think it's offensive to morals and public decency? Let me ask you another question. Is it offensive to morals and public decency to allow people to suffer and animals to suffer with diseases we can get a hand on? You think that's offensive to morals and public decency? Raise your hand if you do. Hell yeah! This thing is going up for vote November 6th. And I can tell you, I'm not sure what's going to happen. I've been there. I spoke to the community. I spoke to high school kids. I spoke to reporters. I spoke to public radio. Wisconsin. It's in Mount Horeb, Wisconsin. And I just got back, and I will tell you, they've got vote no and vote yes signs, and every law, it's an even split. And there's a very good chance it will happen. Now, why are they going to do this? Well, first of all, here's the funny part. It's not really funny. It's revealing, though. The P.O. box of this place is in Mount Horeb. The actual facility is not in Mount Horeb. It's in Blue Mountains or something like this, someplace next door. So when this passes, and I think it may, hope it doesn't, but if it does, it won't actually impact them directly, but fear not, that's not what they're all about anyhow. What they're trying to do is create a crusade. And they've already started drafting county language. And they've already said very plainly in the papers that they plan to bring this to other communities. Well, they only have to go to how many more? 2. And then all of the dogs and cats, and I'm not talking about cat breeders in this case anyhow, are gone from the U.S. research landscape. This incredibly powerful model, because we share so many diseases. They only have to attack 3 and pass these kinds of things. And then, so this is what's happening, and that's how they do it. Here's the other thing they do. So, some legislators are probably so tired of hearing from people right now about this thing, right? And then he says, well, I mean, what kind of impact is this going to have, right? And then somebody else says, well, it's not really even going to impact this Class A breeder because he's not even in Mount Horeb. and that legislator says, oh, well, then I'm sick of hearing about it, just pass it, I'm good. Right, let's put it on the ballot, I'm good, what difference does it make, right? They all say, okay. And that's called low-hanging fruit, and they do this all the time. They go and they collect a bunch of these, and then when they want to go someplace for the big kill, they say, I don't see why you have a problem with this. Here's 15 other places that are already doing this. See?
They don't have it.
That don't have it. They're not doing it, so this is all part of the game, and we're seeing it unfold. It's a bit terrifying because there's only 2 more of these groups available to provide really important dogs for research, and they're a critical model. By the way, the other thing the FDA requires for drug development during that preclinical testing is that after it's evaluated in rodents, it must be evaluated in either a monkey or a dog. So this is actually quite extraordinary. What on earth is happening? Why is this happening? This is why it's happening in this particular case. I think I've spoken to you about this group in the past. There's a group called White Coat Waste. And if you haven't read this report, Spending to Death, you need to look it up online and read it. Don't do it when you're eating. It's extraordinary. And basically this is a group of animal rights extremists who are masquerading as fiscal conservatives. And their whole contention here is that work with animals is just a matter of wasteful government spending, and that the government can save the taxpayers a lot of money if they just stop funding all of this research. They also say, now we don't care about the private companies, and what end are they on? Are they on the testing or the basic research end? The private companies. They're on the testing end. They can do all the research, they can keep doing it, but the government spending needs to stop. Well, the government funds what? All the basic research. And does applied research happen without basic research? No, now you see the problem. And they know this, but the public doesn't. They're like, yeah, let the private guys do it. The private guys don't have enough money to do all the basic research or people to come up with this. So it's quite serious. Of course, what they're telling people is animals are mistreated, that there are non-animal alternatives that can be used to replace animals entirely. And again, that research with animals does not help people. That's what they've been saying. So let's look at these claims in turn. Okay, let's start with animals in research being mistreated. I will tell you first that there's an entire field, it's an entire profession of folks whose job it is to care for research animals. They do not live in labs with researchers. They live in specially designed facilities, in specially designed housing, with air that's better than the air you'll ever breathe in the hospital or a nursing home or your house for the rest of your life, 10 to 15 pure air changes per hour. The folks who actually care for them are specially trained husbandry people, specially trained vet techs, specially boarded veterinarians. What do I mean? I mean that the board exam for these veterinarians is a 30% pass rate. And it takes these people 3, 4, sometimes 5 times to pass. There are less than 1,000 of them in the entire world. They have a lot of information, and 4 of my 7 are ACLAN boarded veterinarians, specially trained behaviorists. Of course, then we have a whole bunch of specially trained compliance people and folks monitoring, and so then of course we have unannounced inspections from external agencies and accrediting bodies and all of this, right? And the reason for that is because great animal care brings great science. You cannot have great science if you don't have great animal care. It's garbage in, garbage out, right? And the goal is to reduce variation. So you want to have the happiest, healthiest animal you could possibly have, so that this information, when it is collected, is extraordinarily meaningful, because we want to reduce always the amount of animals that are still necessary for this work. So what does great animal care look like?
Ever since I was 4 or 5 years old, I remember begging my mother to let me have a dog. I never really thought about scientific background, but the opportunity came along, and that was 26, 27 years ago.
I have been in this facility 15 years now. When you want to work with the animals, you have to be passionate. You have your moral, ethical, scientific responsibility, and you have your own conscience to answer for.
My big passion is animal welfare. In a scientific environment, we need people that are driven primarily by their passion for animals.
When I come in every morning, I know that I'm going to see my dogs, my monkeys, my pigs, my rats and mice, and I enjoy working with them.
Our surgeons are highly trained. They're really good at what they do. When the dogs come in, they're happy, they're excited to see the staff.
when surgical team takes an animal into the preparation suite. It's nice to see the dog happily following her, wagging his or her tail.
Dogs are important in research because there's a lot of things that dogs are well suited to that another species might not be. It makes me really happy that over the years we've been able to introduce adoption as part of our program so that the dogs that have given something to research to help humans, we can give back to them.
I have a dog right now who is really my wingman. He comes everywhere with me. We go camping together, hiking, swimming, we do kite trips, and he's happy to be by my side.
We are contributing a great deal to society as a whole. We're making a difference for people who are sick. We're making a difference for people who need different therapies that aren't currently available. We're also contributing to the health of our pets.
I really believe that what I do here is immeasurable. Just the sheer numbers of animals that I've managed to make things better for, nobody can take that away from me. And I'm really proud for the research community that I was able to do that, and I'm proud for myself as well.
You are benefiting from animal research in every aspect of your life, right from the time that you're born. They are contributing to the development of medications that patients are going to be using, not just for human beings, but also for animals.
When I see people come in and they're animal lovers, and I see them driving what their beliefs are, I think maybe I did do some good. I gave them the confidence to go ahead and not change the way they think or not get used to the way things are, but to keep driving things forward. And it's nice when I can look at a person I've trained that came in, they're continuing that push and that drive.
These are not actors. This is a private company. And those are their animals. And those are the people who care for them in those various titles. This is how animals are cared for in research settings by the lab animal science community. Now, I also am the director of one of the 5 largest research animal care programs in the country, in addition to all these other things that I'm involved with because they're so important. And I have to show you some of my animals just because they're cuter than everybody else's. So, here are some of ours. This was for a photo shoot we did for environmental enrichment record, right? These are guinea pigs, and more guinea pigs, and our folks with the guinea pigs and some of the things that we enrich them with. By the way, when you are doing environmental enrichment for animals, and anybody who works in the zoo community knows this, right? Whenever you keep animals in captivity, you've got to address not just their physical needs, their health needs, but you've also got to address their behavioral and their intellectual needs and their social needs. And so we have to account for all of this if we really want these animals to be as healthy as we need them to be, right? And not stressed in order for this information that is collected from them to be meaningful. And so we spent a lot of time doing that. This is a little lamb, John. He was not supposed to be born in our facility. Nobody knew his mother was pregnant. I got a call on Sunday. There's a baby. I'm like, oh, knock it off. Go back to work. But they're like, no, there is. I said, stop it. There's nowhere. And then I got a picture. So, oh, yeah. We have a baby. Where did they come from? Imagine their surprise when they walked in, but there's a baby. Yeah. So I'll tell you something about sheep, and then they can be challenging to keep in captivity because they can't stand to be alone, right? So we always have them socially housed, 2 or 3 in Iran, and this is pretty typical. But at some point, depending on the study, there may be 1 who's alone, and it's a terrible thing. They won't eat. All they do is bleed and bleed and bleed. It's horrible for them because they're flock animals, right? So what we did with John is we made a companion animal out of him. So we neutered him and we dehorned him when it was time, and then we halter trained him so that he was really good with folks, with people, right? So he enjoyed people because he also can't be alone. And then whenever we had a sheep who was by herself, we put John in there, and it was a love affair, and that solved that problem. And I would say that that's pretty nice treatment, personally. The other thing about sheep is they like a flock. So 2 or 3, well, it does provide social housing. It's not really what they like. A flock is like 5 or 6, and you guys can tell me more than this. But I can tell you that their behaviors changed when we gave them an opportunity to be in more of a flock setting than just 2 or 3 together. And again, our goal is to get these animals to be as steady state normal as possible. So I built them a flock room. And here it is. And they loved it, and this is really cute because they started doing the service sheep thing. And they'd all gather and kind of look at you just like service sheep. And we did some studies to show that they were ruminating more and doing all the other things that sheep do. And we write about this, we present about this, we share this with everybody in research. And it wasn't all that expensive to do, but it made a big difference in the quality of life of these animals. These animals, remember, lose everything for us. And they deserve the best quality of life that we can provide for them. That's just how it needs to be. And so we're always striving to do it better. We took our rabbits. I don't really keep animals in cages. The cages sometimes, depending on what they are, mice are in cages. This is a funny story. Actually, people will say, "Oh, where do you keep your mice?" And I said, "Well, they're in cages." "Oh, well, there's a small little cage." And I said, "Have you ever had a hamster?" "Yeah." "Well, where'd you keep it?" "No, it's a small little box." All right, well, ours have nesting material. Did yours? What's nesting material? All right, never mind. It's just essential for their physiology and, normal behavior. Rabbits sometimes couldn't rear, and they had adjusted the housing standards to allow them to do this, to stand up, and we just took them out completely, and it wasn't expensive to do. We just pulled them out, put bedding on the floor. We had to socially house them, which is the default by law, by regulations, for animals in research. So these are just a couple of cheap, look, that's a garbage can, we cut the bottom out. It didn't cost money. Well, I mean, it cost somebody money. I don't know whose garbage can it was.
Probably cleaner than my garbage.
I'll bet. I'm sure it is. It goes through 180-degree rinse. I can guarantee you it's cleaner than your garbage can. Here's just some more pictures. They get to tunnel. They like to hide behind things. They like to, when we provide this for them to eat, poop in it. And they all poop in that. It's funny, I guess that's why they're so easy to litter train. Pigeons. The space for pigeons is, I think, a bit small. I mean, the guide, they really can be in a box this big. And the guide's okay with that. We were not. They weren't breeding well. So we built little flight rooms for them. Also not hard. The hardest part is finding the right materials because they have to go through these industrial washers. They get a lot of chemicals at very high temps and then autoclaves depending on what they are, right? In order to be really, really clean because we can't have any variability. So the hardest part is finding materials that are appropriate and then being able to clean up. And you see this is a very clean room. You'll see this every day when you walk in this facility. Our folks are trained to do all of these things. So we built them a couple of flight rooms. This is really cool. We gave them some perches so they could do their whole pecking order thing. I'm more important than you, so I'm going to be up here. You're lowly scum, you to be on the floor. They do all of this. And the other thing, so you'll see all of this that you couldn't see in the cages before, right? And you'll hear all these little, like when you're out in the city, ooh, they make all the little sounds. We didn't really hear so many natural anything from the pigeons until we did this. And then someone put a swing in there, and I don't know why they use it because I only ever see them on concrete buildings, but they seem to like it. And the thing that's really funny about it is I said, "Where'd you get that swing? What's it called?" She said, "Oh, it's a chicken swing." I said, "Okay, now I really don't know what else you're on." A chicken swing, okay, never mind. They use it.
You don't put statues in there for them?
You're right, that's what I should do, right? Put a statue in there. Some guy reading the paper. That's what they eat. That's enriching. That's normal pigeon behavior. Yeah, that's good. All that stuff's going in there. When I come back next year, I'm going to show you the picture. Just one more picture I can show you. This is our little Dash. And you don't make fun of me. He had a birthday party last year. He's going to be 2 in January. And we have found him a home. He's going off to live with the flock of sheep I just showed you, who live 5 minutes from my house because they were all adopted. Which is wonderful because I get to see the Greys for the first time and I can visit them whenever they want, and they're going to take Dash as well. This is a really amazing pig. So he was part of a skin study that has to do with eczema and improving treatments, and so he doesn't have to be terminal, right? They just biopsied a few things and they have the results. The researchers are so funny. Pigs are really, as you know, really smart, really social. And so even though the study's over, they come over like 3 or 4 times a week to train Dash to do things because he's real smart and run him up and down the hall. It's really funny. So one goes on one end of the hall, the other one goes on the other end of the hall, and they have marshmallows. And he runs back and forth until he's completely sick of them. And then he just goes back into his room and you never see him again. That's it. I'm done with you. But he knows tricks. He can sit, he can stand, he can heel, and he can push a cart. This is a remarkable thing. And he's going to a new home, so this is great. And Dr. Henry had mentioned yesterday when she started talking about Homes for Animal Heroes that they always had an adoption program, and that's true of most of us. Research institutions have been adopting animals of every kind, including rodents, for over 40 years. But we've created Homes for Animal Heroes for dogs in particular, to help animals be successful in their homes. They have to learn about, traffic noises and loud dinners and stairs and things like this, and also to counter the Beagle Freedom Project's nonsense that was happening. So these are my animals, and you just saw animals from a group of colleagues of mine. If you want to see more because you think we're the only two institutions in the entire country that do this, then I encourage you to go to a website called Come See Our World, and on it you will find hundreds of images of animals in research settings just like ours, with short descriptions about why that particular animal model is important, and also a really easy way to just click Facebook or whatever and share them. And if you follow me on Facebook, you'll see I put those up from time to time. The other thing that's really great about this is we encourage the media to come here. You can take any of these images yourselves through a Creative Commons license and use it. And so whenever we integrate with the media, we offer them the opportunity to go take images from here. They are representative and accurate, unlike the grainy nonsense that's in black and white or horrific that they've been using for 30 years, right? To be honest, they never had a place to get good images, accurate images. Now they do, so everybody can use them, school teachers, everyone. All right, so I think we have taken care of the animals are mistreated piece. Anybody have questions? How many of you feel better? Because you weren't quite sure, raise your hand, be honest. Yeah, good, I'm glad. Okay. What is this alternative stuff they're talking about? Where we can replace animals with something on a chip kind of thing. And this is primarily what they're talking about when they're talking about it, because it's the new hot thing, it's the new hot buzzword. And they know when they say stem cell technologies and organs on chips, that nobody in the public knows what the hell they're talking about. So surely they must know, these people who are telling them, because they're using these impressive terms, right? And they must know what they're talking about, and people believe it. So just to give you a quick, very, very basic description. You can seed things with human cells that will then differentiate to become certain things. In this case, this is a model of a lung on a chip. So they put somebody's cells on there, they made it act like lung cells, and they couldn't really get them to do what they expected them to do until they put these things on the side, these vacuums on the side, which cause things to contract, expand and contract. And when they did that, because that's what the lungs do, they started to see some of the same function. So it's really quite interesting, and these things are in development, and the irony here is that the private companies who were really pushing the development of these technologies started with the pharmaceutical companies. It is far cheaper to do your preclinical testing on this than it is to maintain animal colonies. I just told you about all those people and all those fancy places they have to live in, and all of it. I mean, it's expensive, really expensive. And so they're sort of pushing this. This is what one actually looks like. It's kind of cool. Now, if you're going to do a preclinical test, and then you're planning to go put that into a human being after that, you can't just look at one organ system in most cases. Maybe in some you can, right? You need something that can act like a human on a chip, and people are still working on that. And these technologies are being developed in Japan, in Germany, in the US, maybe a couple other places, but that's where the big stuff is happening. And then the hope would be then, in that case, if they can do this, that we can get to this sort of place, where we have our basic research, that's for now anyway, going to involve animals, right? We can move to some maybe of these human-on-a-chip technologies or organ-on-chip technologies, get our preclinical information from that, right? There's something else that can happen here. If I take tissue from sick person A and see their cells on here, and then I test a variety of drugs on that chip, I now know exactly which drug person A is going to respond best to. It's sort of a personalized medicine thing for that. It's kind of exciting, very exciting. And then after that, then I can go ahead with my human clinical trials and eventually. And so when they're talking about this organ on a chip and human on a chip stuff, they're really talking about using it in this preclinical place. They don't say that because they don't even know what they're talking about, because they don't understand that first graph I showed you, right? So they just throw these terms around, but they don't know. I will tell you this, one of the gentlemen who's working on this very, very hard is a guy named Uwe Marx. He's in Germany, and I had a chance to interview him because when I was the president of the Association for Laboratory Animal Science, I got to choose my meeting theme, and it was integrating non-animal technologies into our work, and that was the theme. And so he was my keynote speaker, and he came, and the bottom line is he's working on human on a chip like others, and his prediction for when this might actually take place and be able to replace most, if not all, animals is 2035. So the technology is in development. There's the regulatory agencies are sort of a holdup because they have to have proof that this is going to work as well as the animal data they've collected in preclinical testing, right? And so the irony here is that what they want these folks to do is to create animals on chips so that then they can actually look at the data they get from animals on chips and compare it to all the animal data they have. And if those two things correspond, then that may be compelling enough for them to say, okay, we think this will work then as an option for preclinical testing with human tissue before you introduce things to human beings. This is very exciting. When I was in Wisconsin, of course, all the kids were saying this, and all the people were, well, what about all the alternatives? What about the alternatives? I heard there's an alternative. And I gave them some information, but I've taken to, similar to the farm babe here, I've taken to sort of leading them down their own path of inquiry, right? And so I asked these kids, and I said this to them too, so I challenge you, when you're hearing this from people, to ask them some questions. What's this new thing called that they're using for preclinical trials? Which company makes it? And when did the FDA approve it? And when they go look, they're going to find out there's no such thing. Because there's no such thing. No such thing. So this is where we are. But I'm a big fan of this. This is one of my mantras, stronger science, faster cures, fewer animals. This is the way to get to fewer animals. In fact, the animals are helping us remove them from the equation. The more we know, the more clever we are, the more we can develop these new technologies, the less likely we'll have to rely on them. At least on the testing side of things, maybe first. But maybe as we know more and more, we might get down to the basic research end of things and be able to use these technologies to do that too. So now the third thing they said is this stuff does not work, it does not translate to human cures, and they get that and they say all the time, and that's because 95% of the drugs that work in animals don't work in people. First of all, that's misleading because we never evaluated whether or not the drug worked in animals. Remember that? We want to know, is it safe and does it go where it's supposed to? So it's a whole crop. But it is also true that this is right. 95% of the drugs that are screened in animals and people, by the way, over 80% of those that are tested in humans also don't make it to trial. I don't know why they're not telling us about that, right? They say 95% that are tested in animals don't make it to the public because animals are a bad model. But when you tell them over 80% of those same drugs don't make it in human clinical trials to market, they never say, well, that's because people are bad models. They just ignore that entirely. But it is true that really basically 5% of them get out there. And each one costs about $1 billion to make. This is actually an NIH website which discusses this. There are countless others. But in this particular case, what they're doing is those drugs that didn't make it are on a shelf somewhere. And now that we have more basic information coming from basic research, we're learning more and more about mechanisms that these various drugs on the shelves may actually impact. And now people are starting to repurpose drugs. Like, wait a minute. That guy that didn't work for, I don't know, make something up, liver cancer. That drug that didn't work for liver cancer? Well, now that I understand the mechanism of this other thing, it might work for Alzheimer's disease. I'm making this up, but this is the kind of repurposing that's happening, and that information is coming from basic research. That's how critical this is. Okay, but what is really happening, right? So what they're saying is that this is true because animals are so different from people. And that's why they don't translate. What we find out in animals does not translate to humans. Now we know this is not true because evolution argues against it. And there's a huge amount of conservation of biological systems across species. We know this for a fact. This is science. It's not debatable. We know this, right? So then what is maybe potentially contributing to this? And I will tell you what it is, and the scientists in the room won't be happy about it, but they know. There's a lot of work now suggesting that the rigor with which our researchers in this country, not just our country, but other countries too, the rigor with which they do science needs to be reevaluated because they have, in the continual pursuit of publication, because they can't survive without it, they sometimes don't design their studies as carefully as they should. Some things are listed as significant in papers, and later on someone does a meta-analysis and finds that there's a little bit of bias there. There's, for example, you're looking at a particular thing, the stats don't say that that thing is significant, so you look at it a different way with a different calculation. Oh yeah, it looks significant. Or you tell your story a little differently. And this is not because researchers don't care about what they're doing, it's a culture. It's a culture. The other thing that doesn't happen very often, there's a little bit more of it now, is that if you test something and you don't find anything, that's not usually very interesting to the people who are going to publish it. They want the findings, the positive findings. But the negative findings are just as important, right? Don't you think it's just as important to know what isn't true as it is to know what is true? Yes, especially if you're the drug company now who's going to be getting all this information and try to create that new little drug, right? If you're missing a whole bunch of what isn't true, you don't have all the information you need to start making your drug, which is another reason why They don't all make it to the end of the, okay? So there's a lot going on here, but it has very little to do with a lack of correspondence between the biology between animals and people. Just so you know, because I don't mean to be upset about this, there are initiatives in place helping researchers now, especially young researchers, sort of standardize their practices, make sure that you're not missing any important steps, right? Standardize things, and also make sure that you report everything. For example, if you and I do the same study with mice, and my animals live on corncob bedding, and yours live on cellulose bedding, we'll probably get different answers, depending on what it is we're looking at, right? And so all of these things have to be accounted for, and that kind of stuff has generally not been included in papers. Some of that's changing because of the microbiome. We're not going to get into that. Let me ask you this question, though, before I leave you. All of that aside, 95%, does the 5% that does work matter? Yes. Everywhere I turn, I find people living with cancer, living with diabetes, living with HIV/AIDS, people who would have died before. Does it matter? Yes. Ask him if it matters. He's about to have brain surgery that may save his life. Ask her if it matters. She's being treated for cancer. We heard all about the One Health Initiative yesterday. And now I'm going to leave you with this.
First tested and successful in dogs, we have remarkable news tonight concerning a groundbreaking treatment developed at UW Medicine and now being tested on children. The first results have just come back involving children with a rare, deadly form of muscular dystrophy. Dave broke this story last May, and you really have an exciting update.
Yeah, it is exciting here. Last year, we broke the story of how gene therapy has reversed the deadly disease in dogs. While still very early in clinical trials, it's now showing similar success in kids. They are the moments of unbridled joy.
Yay!
For children whose lives can't be taken for granted.
Can you say hi?
Parents soaking in those precious moments and praying for a miracle.
You swimming? Kick, kick, kick.
For the past 10 years, researchers at UW Regenerative Medicine have been trying to find a cure for myotubular myopathy, a rare and deadly form of muscular dystrophy.
The clock is ticking for a lot of kids out there, though. It is. I'm painfully aware of that.
We're gonna move ya.
It is a continuous hum of beeps and breathing machines inside this Des Moines home.
All right, here's your air bump.
A constant drip of food to keep Chris Bowers alive.
Yeah, I did.
And a steady dose of Legos to keep him entertained.
He really has been a rock star.
After 9 years of defying the odds, his parents are hoping for a miracle.
Cautiously optimistic. It's hard to imagine because Chris, this is what we know.
The same disease affecting Chris also affects dogs.
Very rapidly, over a period of a few weeks, certainly by 4 months of age, the affected puppies are unable to walk. They're very, very weak, and unfortunately they require humane euthanasia.
In an effort to save the lives of dogs and ultimately the lives of children, researchers at UW Medicine infused a replacement gene into these puppies that have myotubular myopathy. These are 5-month-old brothers. The puppy struggling has been given saline. The active pup was given a single treatment of gene replacement therapy and now shows no signs of the disease.
This works remarkably fast in dogs.
Yes.
How fast? Unbelievably fast. So we see effects within 2 to 3 weeks. Good boy.
And here are 2 other dogs treated 4.5 years ago that are now flourishing.
These dogs would normally die around 2 months of age, 2 to 4 months of age. So because of the gene therapy, they've survived now to over 4 years.
Good boys. That's fantastic. That's great. I don't like to use the word a miracle, but it's pretty darn close.
Is it the most profound thing you've ever seen?
No question about it. Absolutely. Yeah. Been at this a long time. I've never seen anything like it.
Last September, the first clinical trials began in 4 boys under the age of 5, all with myotubular myopathy, 3 of them given the same treatment as the dogs. Today, for the first time, those results are public.
That it matches very closely what we saw in the dog study. That study showed that only after about 2 weeks, or a matter of weeks, we saw dramatic improvement in the dog's function, their muscle function. And that appears to be the case in the clinical trial.
Biotech company Audentis says the results have exceeded our expectations. Audentis reports one child attained several age-appropriate developmental milestones, including head control, rolling over, and sitting unassisted. The UW Medicine researchers behind the therapy are thrilled.
Yeah, highly encouraging, better than I'd hoped for.
We were very hopeful, but we were not really surprised. The dogs had the same response that the children are showing right now.
Jane, you want more kisses?
In the months to come, more children will be given the gene transfer therapy. The hope is that the lives of these beautiful kids will one day be transformed, just like these dogs.
When we talked in the past, you were very optimistic that this would work, very hopeful that it would work. Are you just as optimistic now?
Absolutely. Yeah, yes, no question about it.
What do you say to those families who have children with myotubular myopathy? What kind of hope can you give them?
I think we can give them a lot of hope. We're seeing results that restore function to near normal levels. So this is not going to be a partial improvement. We're optimistic that these patients will be able to play and run and behave like normal children.
Do you want more kisses? More kisses? Oh! Oh, mwah, mwah, mwah.
Again, this therapy is still in early clinical trials, but the results are very encouraging. Eventually, the data will be given to the FDA, and they'll seek approval of that drug. Hopefully, all things will go smoothly here.
Dave, we can only imagine families, when you see those kids, are willing to do almost anything to have their child get this treatment, which must be wildly expensive, I would think.
Yeah, it is incredibly expensive. It requires both private and public research dollars. Regenerative medicine is groundbreaking. The vast majority of rare diseases are caused by defective genes. This is a technology that can replace those defective genes.
And something else that I'm sure some of our viewers are thinking about, this involves animals, and there's always a lot of questions when we talk about and show animals in this kind of experiment.
Yeah, it's a sensitive issue. I'm sensitive to it. This disease is really a naturally occurring disease in dogs. That's important to know here. As we saw, this treatment is saving their lives as well and could offer advancements in veterinary medicine. We will see. This is a very exciting time in the world of medicine, but again, it requires a lot of money.
And a good deal of it is happening right here in the Pacific Northwest at UW.
Absolutely.
Dave, it's a great story.
Now, how many of you think that 5% matters to those children and their families? Yes?